Hello everybody and welcome. Here we are, it's 3MT time. It's a moment we've all been waiting for because a lot of people have put a lot of effort into the fantastic presentations you're about to see today. We're streaming live, as you might have heard, and so we've got an audience at home who's excited to be here and to see all the presentations. We've got a huge audience in the room who'll be sharing lunch with us afterwards to celebrate. Although only one person will win, it's actually fair to say that every person who's being presented today is actually already winning, and let's just admit it, we're all winners because we got here, we're at a live event, we're doing blended, we're streaming as well, so it's just so exciting. But let's begin by taking a moment to just acknowledge that we're taking on a tradition from the Indigenous elders of this land, just every time we come together to stop, to notice one another, to ground ourselves in all of the knowledges that are held here, despite the fact we are a little above the ground right now. The knowledge is deep here. And as people who are interested in the pursuit of knowledge, it's so important that we take this time to recognise that different people bring different knowledges. And so when we come together, what we need to say is respect. It's very important first that we respect the Indigenous elders of this land. Here we are in Wurundjeri country in the Eastern Kulin Nation. So respects to elders past and present. Also to any First Nations people who are in the room, respect to your traditions. And that extends to us, to respect one another's beliefs. And in the nature of science, if we want to get technical about it, that we would actually also be aware of the ontological and epistemological beliefs that we all bring to our research and how different they are from one another. And so this tradition is established so that we can pay that respect. Firstly on these unceded lands, but also to one another. So as we move forwards, I would like you to take that sense of depth of knowledge that are in the lands that we stand on and proceed to be excited by what contemporary knowledges have been discovered in just the last year. Since 3MT is a celebration of PhD research. It's an international competition that people from all around the globe participate in, and Melbourne University is very proud to be hosting the grand final of the 3MT presentation, celebration here today. So thank you to all of our presenters. I believe this is all of you sitting along here, am I right? Thank you. It's really exciting to have your work. And what we've learned through years of lockdowns and pandemics is that we can very easily create videos from home. And so today we're going to be having video presentations, pre-recorded video presentations of the final. Oh, I won't start singing just yet. There may be singing later, but not just yet. And so for you, this is slightly less stressful, slightly less anxiety provoking than it would have been if you were sitting here going, I'm number 8. That means I have to listen through all these brilliant people before I get to do my job. But this is the thing about the University of Melbourne. We're all brilliant people. When you come and you join the research community here, you find yourself surrounded by equally brilliant people. And often you've come from places where people point to you and say, you're the most brilliant, you're outstanding. And then you join us here, and we're all outstanding. And that doesn't detract from anybody's unique outstandingness, by the way. There's room for all of us here. And so the Researcher Development Unit is the host of this event today on behalf of the University of Melbourne. And the Researcher Development Unit, which I'm honoured to be the director of currently, is a unit that's designed to empower researchers across this vast ecosystem of our research community, to recognise that it can be intimidating to be surrounded by people who are equally brilliant to oneself, and that we need to do professional development work to help us to continue to make our unique contribution to the generation of knowledge, because ultimately we want to have an impact. The university is on a mission to make a difference in the world, and each one of us contributes to that by our own piece of research. And PhD research is the most fabulous of all because you concentrate on a single task, a single project for such a long time, and yet by the end, you can put it all into 3 minutes. It's kind of remarkable, but it's an experience of being humble and being humbled, as well as being magnificent and discovering something altogether amazing. So that's what we're here to celebrate today. And the ability to put that into 3 minutes is really important too, because you're generously giving me about 5 or 6 minutes of your time, and even that might be feeling quite long because we move pretty fast in the world at the moment. And so to be able to describe the impact of your research in 3 minutes is an extraordinary task, but it certainly has been done in the videos today. So we can get excited for that. But first, I'd really like to take a moment to welcome our very exciting group of judges who I personally have only one sentence to say about, but I would have liked 3 minutes to be able to describe each of their contributions, because we certainly do have an astounding group of people here. Mark, I'll start with you. So Mark's a senior lecturer in information systems and particularly in digital ethics. He's interested in the intersection of technology and philosophy, and he's active in science communication. Thank you so much for being here today. Clem is a published author, cultural critic, autistic advocate, and screenwriting researcher, currently completing a PhD at RMIT, and I understand it as being at the intersection of autistic identity and screenwriting, so a fantastic topic also. You're getting the vibe, right? Cool people down the front here. Cassie is our last year's winner. Everybody, Cassie, last year's winner. That is really an amazing experience. So you know how everybody's feeling right now. But Cassie won last year for her presentation titled More Than a Place to Wait, Untapped Potential of Hospital Waiting Areas. And I believe you're a physiotherapist, I'm right, aren't I? Yes, and Cassie's now doing your postdoc at Alfred Health. So great things come after PhDs. Sometimes they're postdocs. Sometimes you wander off into the real world and make a difference out there. Sometimes you join our professional staff and make a difference from within, but at a different role. Sometimes you go into government. And sometimes you stay hanging around in universities for your whole life. It's so many options available to us. And Anthea, who is a dear colleague of mine who I've been fortunate enough to do quite a lot of research with, but Anthea is currently a Mackenzie postdoctoral fellow, an ethnomusicologist who specialises in, disability music culture, military music, organology, which is not about the internals of the body, it's the internals of instruments, FYI, and archiving. Incredibly multi-skilled, amazing human being to have on a research project in my experience. So maybe a round of applause, if we could, for our wonderful judges. Terrific. So without further ado, let me introduce you into how we will flow. For the next half hour, we're going to be watching the screen. Now, I know we all love watching our screens, so that should be nice and comfortable for us all, and we know that we can get into certain positions. Make sure that you are not uncomfortable. If you've got your legs crossed, consider placing them down on the ground in front of you, all those kinds of things, because we sit at our desks way too much, right? Probably everybody in this room. So just take care of yourselves. But we will be actually moving really fast. So that will suit us all as well, because we'll have people's names are going to come up on the screen, their titles, and then we'll launch into their presentations. Your job is to reach your own personal opinion about which of these amazing presentations you personally consider to be most impactful. The truth is that they all are. That's life in this fantastic university. Every single person is always pretty extraordinary around you, but you can still have a preference. So at the end of this process, your preference, you'll be scanning a QR code, and people at home will also have access to that, and you will be voting on the People's Choice Award. In the meantime, our judges will exit the room, they will deliberate and come to a fair and just conclusion about who today is going to be our winner, and then we will celebrate with lunch afterwards. All okay? Thanks so much, everybody, for being here. It makes such a big difference to everybody up the front, and it makes a big difference to us to know that the community is interested to see what impact our newest researchers are having. So, without further ado, is it all right if I begin? Yes? Getting down off the stage.
One avenue for increasing the livability of cities is to plant more trees. More trees means more shade, cooler cities, and more opportunities to absorb and retain carbon dioxide. Urban trees also enhance spaces for people to connect and reset and provide valuable habitat for other urban dwellers like birds and insects. I'd like you to take a moment now to think about the trees on the street that you live on, or perhaps a nearby park or reserve. Can you picture or identify those trees? And do any of them happen to be edible? Productive or food-producing trees might not be what first comes to mind when you think of urban trees, and that's exactly where my research seeks to contribute. As a starting point, the literature tells us that local governments tend to be wary of planting food-producing trees due to pests, mess, and other risks, but that communities around the world continue to experiment with these trees, planting them in verges and nature strips as part of food forests or urban orchards. Therefore, in my project, I'll be investigating practices, what people actually do, the relationships, the connections and meaning made between elements, and the narratives, the stories and ideas that relate to urban food-producing trees, in order to better understand the way in which they can contribute to the livability and sustainability of cities. In order to do this, I'll be interviewing local groups within metropolitan Melbourne that are actively planting food-producing trees, and I'll also be talking to the local governments where these activities are happening. I'm in the early stages of my research, and most of the interviews that I've done have been with local government representatives. Therefore, it's too soon to draw any conclusions from my conversations. Rather, I'd like to share two insights that shine a light on some of the thinking and ideas that my research will generate. The first of these is the conversations I've had about the different ways that urban food-producing trees have been and can be managed, the formal and informal processes of co-management. These conversations suggest further thinking is needed about the way urban trees can be jointly managed with communities and local governments. The second insight is that local governments typically don't see food-producing trees as high on the list for achieving objectives of canopy cover and urban cooling, but they are open to these trees when there is high community demand and when multiple benefits can be seen. This suggests that a more holistic approach to thinking about the role of trees in the urban landscape might be necessary. Through further interviews, observation, and document analysis, I hope to better understand the processes by which communities and local governments sense and decide what trees belong in our landscapes, and the skills and governance approaches needed in order to care for and live and thrive with those trees.
Location, location. As the daughter of a real estate agent, I learned from a young age the importance of choosing where to live. Is your house close to a train station? Do you have good neighbours? Is it walking distance to a decent coffee shop? All of these factors can contribute to where you live and how long you live there. For our immune cells, location is equally important. In our bodies, we find immune cells in draining tissues known as lymph nodes, which are a hub of immune activity. In these lymph nodes, immune cells are organised into distinct compartments, or neighbourhoods, which ensure immune cells have the correct neighbours and environment to allow them to respond appropriately and efficiently to infection and disease. In my PhD, I looked at the role lymph node location plays in the maintenance and function of immune memory, specifically memory T cells. Memory T cells are a white blood cell which provide us with long-term immune protection against different infections and also play a key role in fighting cancer. Memory T cells are what we hope to generate when we vaccinate against viral infections such as COVID-19, as these special cells can live in our lymph nodes long-term and prevent us from catching the same virus over and over again. However, many of our current vaccines fail to generate a strong memory T cell response. We believe that if we can understand exactly where memory T cells live in our lymph nodes, we may be able to better generate immune cell memory following vaccines and immunotherapy. However, the exact place where these elusive memory cells are found in our lymph nodes remains poorly understood. In my research, I asked two questions. Where are these crucial memory cells located in our lymph nodes, and who are their cellular neighbours? To address these questions, I consider myself a bit of a lymph node photographer. I use fluorescence microscopy to take photos of lymph nodes following viral infection to look at exactly where these memory T cells are found. In doing so, I've discovered that they actually live in a very different region of the lymph node to other immune cells, closer to the portals of entry and exit, or the lymph node train stations. I've showed that this location was conserved across various infections, and interestingly, this neighbourhood deteriorates as we age. I've also described a range of cellular neighbours which interact with our memory T cells in this very important neighbourhood. I hope that in teasing apart the interactions within this memory suburb, we can better understand how to build and maintain immune memory, what happens when this neighbourhood starts to fall down, and how we can create a better environment in our lymph nodes for our immune cells to live long term. Ultimately, superior immunity may all come down to finding the right location. Thank you.
Sick with a bacterial infection like tonsillitis or pneumonia, we go to the doctor and the doctor gives us antibiotics. We take these antibiotics and then we get better. That's the way it's worked for decades. However, just as soon as humans began developing medicines to combat infections, bacteria started evolving new ways to defend themselves. Instead, you could now be faced with an infection resistant to all known antibiotics, leaving the doctor powerless to help you. Rates of infections caused by resistant bacteria have been steadily rising for decades, and by 2050 it's estimated that every single year, 10 million people will lose their lives as a result. One of the best ways we can defend against resistant bacteria is the rapid detection of outbreaks. This allows us to isolate patients, limit the number of people who become infected, and thus save lives. So how do we detect outbreaks? Well, like all good detectives, we use fingerprints. And in the case of bacteria, their genome is their fingerprint. It works like this. Swabs are taken from the scene of the crime and infected human, and we genetically sequence the bacteria that we find. Using computer programs fluent in the language of the genetic code, we can compare the genomic fingerprints. Then, based on the number of differences between them, we can begin to understand which bacteria are closely related and which are not. If there are multiple bacteria that are closely related, we can be confident that transmission is occurring and an outbreak is in progress. Mystery solved. However, as in all good mysteries, the culprit is not always easy to find, and sometimes the evidence can point to multiple suspects. Although advancements in technology mean that it's now relatively easy to sequence genomes, the best way to compare them and identify transmission events is not well understood. My research tries to fix this problem and aims to develop a new method that accurately and reliably detects outbreaks of resistant infections using genomic evidence. To find this new method, I looked at outbreaks that had already been solved. Using a comprehensive dataset collected over the course of a year that contained both genomic data and data detailing patient movements within the hospital, I compared different methods of interpreting the genomic evidence to see which one was best at correctly identifying the outbreaks. Using this framework, I developed a new method that identifies outbreaks faster and more accurately. It can be used to detect outbreaks in close to real time, allowing us to stay one step ahead of the bacteria. It's also standardized, meaning that genomic detectives all over the world can use it, confident it will solve their outbreaks too. Use of my new method will allow us to target infection control resources where they're needed most and stop outbreaks of resistant bacteria before they spread. The best cure for a bacterial infection that has no cure is not getting infected in the first place. The outbreak detection techniques that I have developed can go a long way to prevent infections, stop the spread, and ultimately, save lives.
Over 60% of the population over the age of 40 suffer from a particular form of dry eye disease. With dry eye disease, you experience redness to the eyes, sore and burning sensation, as well as glare and blurry vision. Lots of research has gone into investigating the different causes of dry eye disease, but scientists in the optometry community may have overlooked the relationship between the immune cells and the meibomian glands in the eyelid. The immune cells in other parts of the eye are fairly well understood. My PhD is the first to look into the immune cells in the meibomian glands in the eyelid. But first, let me take you back. How excited were you when we first got out of lockdown? I was so excited to finally reconnect with my friends and family and feel a sense of freedom again and feel like things were finally getting back to normal. There are certain immune cells that feel the same. Immune cells like to work together with other immune cells to look after their home. A specific type of immune cell is the dendritic cell. The dendritic cell is the focus of my PhD. Dendritic cells' role is to help protect the eye from any infections. They also help the eye maintain healthy functions. When they're happy, they like to work with other dendritic cells to look after their home. When they sense a state of emergency, though, they're in suffering. They hide, self-isolate, stop connecting with their friends, and eventually stop looking after their homes. In the eyelid, the dendritic cell's home is the meibomian glands. We have more than 20 meibomian glands all along our eyelid margin, very close to our eyelashes. They line up like houses along the street. Their role is to produce oils that are part of the tears to help the tears from drying out too quickly. With dry eye disease, the meibomian gland shortens and dies away and stops producing oils. My PhD is focusing on looking at the role that the dendritic cells have in looking after their homes, the meibomian glands, and determining if their happy and suffering states are involved in the causes of dry eye disease. By understanding their relationship, we may be able to determine a way to ensure that these dendritic cells and the meibomian glands live in harmony and they're always in a constant, sustainable community. In understanding this, we may be taking a step closer to locking in the tears in the eyes and locking down the causes of dry eye disease. Thank you.
One experience likely shared by all of us at some point is the sharp pricking pain of a paper cut. Now, this paper cut was probably there one day, gone the next, and you probably didn't give it a second thought. Now, while you may not have realized it, your body just underwent an incredibly complex process known as wound healing. This means that multiple cell types in your body, your blood vessels, your nerves, even your hair follicles, all decided to work together in perfect harmony and in just the right sequence to help close this wound. Now, this process works seamlessly when we're younger, but the older we get, it gets a bit trickier. Our cells are less responsive, slower to heal, and in patients with conditions such as diabetes, a small injury such as a mosquito bite can stay unhealed for months, even years, progressively getting worse, with often the only option left being amputation of the affected area to stop the wound from getting worse. Every 30 seconds around the world, someone is losing their arm or their leg due to a chronic wound. So, what can we do about it? We know from decades of previous research that platelets are small fragments in the blood that are very, very important for the first stages of wound healing. As soon as there is an injury, the platelets in your body detect it and they get activated, which means that they go from their typical round shape to growing these long arms out, reaching out to other platelets to form a clot and stop the bleed. And in more recent years, scientists have discovered that platelets are not just important for clot formation, but that when activated, they release millions of little packages into their environment, filled with growth factors and nutrients, essentially sending instructions to the other cells to get the healing process going. And so my project for the last 3 years has been to understand whether we can isolate these packages and use them as an effective treatment to heal chronic wounds. And my results have shown that these packages help skin cells grow twice as faster and make them more mobile so that they can travel to the areas where they're needed, and even help grow new blood vessels and reduce scarring. These findings suggest that this treatment could offer a real solution for patients in the future when there hasn't been one, because no one, no matter how young or old, should have to lose a limb from a paper cut.
I have black hair, 2 eyes, and if you look closely, you might be able to see a scar here. This is what most people see when they look at me. Some people, though, see this. Age-related macular degeneration, or AMD, is the leading cause of irreversible vision loss worldwide. 1 in 7 people with AMD progress to the late stages, where it takes away the ability to read, drive, and see faces. And while there's treatment for some of them, frequent injections into the eye which are not pleasant, we don't have a way to stop or even slow down progression. Knowing who will progress, who that 1 in 7 is going to be, is still a significant challenge. We know that some features of AMD are associated with progression, but we still don't have the full picture. If you've gone to an optometrist before, they might have taken a photo of your eye and said everything looks healthy. They don't say that to people with AMD. These people have drusen, deposits of waste that build up at the back of the eye. On photos, we can see how big they are and how many there are, but not much else. But now, with advanced ways to image the eye, including high-resolution cross-sectional scans, we can see what drusen look like on the inside, like how reflective and uniform they are. And we've learnt that what was previously considered to be all the same thing on a photo is actually many different types of drusen on a scan. To learn more about these new features, I need to look at a lot of scans. I have images from 140 people with AMD, taken every 6 months for 3 years. At baseline alone, I have almost 14,000 scans, and so I spend my days identifying and then quantifying different types of drusen. And because I know who ends up with late AMD, I've discovered that some drusen, like those with big, black cores, are more predictive of progression than others. This is important for 2 reasons. 1, for the people who need injections, the quicker they get treatment, the more vision they retain. People with high-risk features who are more likely to progress need to know this, so they can be vigilant about recognising changes to their vision early. And 2, this expands our understanding of AMD and informs what we should prioritise as researchers. Knowing more about the disease gives us the best chance to stop it before vision is lost. One of my patients with late AMD frequents the same grocery store that I do. She always tells me that if I see her there, I have to say hi because she won't be able to recognise me. And this makes sense because this is what I look like to her. So sometimes when it feels like all I do is look at scans on a computer, I think of her. What I do is a very time-consuming, repetitive, tiny part of a much bigger picture. But the bigger picture's worth it. Thank you.
Having children of our own is a dream so many of us have. But in reality, it's something that many people struggle with, as 1 in 6 couples now have to go through IVF in order to conceive. One major factor that is contributing to this rapid rise of infertility and other reproductive diseases is our exposure to chemicals in our environment that affect our hormonal system, known as endocrine disrupting chemicals, or EDCs for short. These chemicals can mimic and interfere with the natural hormones in our body, like estrogen and testosterone, which can then impact our fertility and reproductive development. So these chemicals are everywhere in our environment. They're things like BPA, which we find in plastics. They're in herbicides and pesticides. They're in cosmetics and medicines. And they're even found naturally in some food products. But the specific one that I research is called DES. Now, DES was a drug that was prescribed to millions of pregnant women worldwide, as was said to help promote healthy pregnancies and prevent things like miscarriage. But this didn't happen. Instead, what we saw in the children that were exposed to DES was a decrease in fertility and an increase in reproductive disorders. But now, what we want to know is, can these effects that we see in the DES children persist into the grandchildren and great-grandchildren of these women? So to answer this question, I exposed pregnant mice to DES and breed them out onto the third generation, or the great-grandchildren. And what did I see? Well, as we saw in humans, that first generation has a decreased fertility compared to our untreated mice. But alarmingly, this decreased fertility persists into the second and third unexposed generation. I also saw that these descendants would hit puberty significantly earlier, and that they had higher rates of disorders that affect the reproductive tract. Therefore, that one ancestral exposure has led to a multitude of effects in the first, second, and third generation. So what can we do with these results? Firstly, there are over 50 million people that have been impacted by DES, so they can use this information to inform their GP of their risks and help lead to early detection of reproductive disease. But what does it mean for the people that haven't been exposed to this drug? Well, as I said at the start, we are continuously exposed to other EDCs in our environment which may have similar effects to DES. So this can help regulate policy around our exposure to these chemicals, to have more stringent rules around new chemical development, and to educate people who are trying for a family to be aware that what they've been exposed to today may not only affect their child, but future generations to come. And then hopefully we can help slow down the rate of infertility in our society so you don't have to struggle to have a family too.
What language did you first learn as a child? Was it English? Was it Somali or Arabic? When you started school and spoke with your languages, were you silenced? Were you told you have to speak English in Australia and that other languages are rude? Like Birhan. Birhan is an African-Australian child born in Melbourne. When I asked him to show me his languages in colour, he told me his story about speaking Tigray, Arabic and English with his family, about speaking a little Arabic in red with his friends, but speaking English in green everywhere else, in his community, at his school, the world. He told me that he wasn't allowed to use his Tigray and Arabic at school because English speakers wouldn't understand him. Birhan, a small black child of refugees, being told that he would exclude white English speakers? What's behind that story? My research looked into the multilingual lives of African-Australian children like Birhan and their experiences at one primary school. I interviewed teachers, parents and students, hoping to find how schools can nurture languages. Instead, I found stories told by teachers, by parents and by students, and these three sets of stories were situated in harmful social stories about African gangs. Within this context, the teachers' stories reflected race-based assumptions. The well-intentioned white teachers told stories about African mothers not talking to their babies until they are 2, about African children starting school with no language, and about older African children using Somali and Arabic to exclude others. The children's counter stories challenged this monolingual mindset. Leah said her 3 African languages are part of who she is. Abel said, I have Eritrean, which is on my heart because it is a country that will stay with me for a long time. Yet none of the children I interviewed complained about speaking only English at school, acquiescing to the school's language expectations. Like the children, the parents also told stories about preserving their own languages, yet the teachers' stories overlooked these realities of the students and their parents. All around us, there are stories about race and perpetuating beliefs about language and culture. A critical race perspective helps us see that. The findings of this study are a call to schools to support Birhan's multilingualism, support his English, but let him speak Arabic, connecting him with family, place and identity. And to white teachers like me, the study says notice the stories that we tell about race and language and listen instead to the stories of students and their families.
It's 1890, and a young girl is really sick. She's been unwell for some time, living with an uncertain future. Her parents, shrouded in the shadows, are worried. Will she recover, or is she approaching the end of her short life? Despair fills the air. Her doctor sits anxiously by her bedside, deep in thought. Unlike the 1890s, we now have more medical treatments available to use. We can support her breathing, her circulation, or any other organs that might be failing. Her doctor is not just thinking about what else can we do, but what should we do, and how will we make such impossible decisions? In the past, paternalism was the accepted approach. Her doctor was responsible for making these treatment decisions. But today, these decisions are meant to be shared between her doctor and her parents. How do we prepare parents to share in this decision-making process? And what does this sharing actually look like? I've answered these questions in my PhD by interviewing paediatricians about their communication approach in this scenario, both in the hypothetical and also following a simulation where medical actors played the role of parents. I found that paediatricians feel responsible to lead decision-making at these times. They make treatment decisions based on what they think is right for the child, but they aren't explicit about this to the parents, and instead skillfully use their communication to help guide parents to make treatment decisions that they think are right. They do so to help parents feel like that they've shared in this decision-making process. And they justify this approach as protection of the child from the harm of invasive treatments and protection of the parents from the burden of making such impossible decisions. So in essence, parents are getting information, but they're not getting true choice about treatments. How parents perceive this communication needs further study, and we need more ethical thinking about whether it's really okay.
Jack is an 85-year-old man. His wife passed away 10 years ago. He lives alone. He has no friends or siblings. He lives in a rural area where he has no one to talk to. Jack is really socially isolated. There are so many people around the world who are just like Jack, are experiencing the issue of social isolation and loneliness. How can we use technologies to help people like Jack to engage in meaningful activities and have a meaningful life? Advancements in technologies have created a lot of opportunities for people to engage in different activities. Through technologies like video conferencing tools, social media platforms, virtual reality, Jack's life is not restricted to the area he's living in right now. He can go to an online lecture in the morning, visit a virtual exhibition in the afternoon, and have a nice reading group discussion after dinner. He can even create his own digital content and share it on social media and become a popular internet influencer. Does that sound challenging? Yes, it can be difficult, because people like Jack may not have enough digital skill, have limited access to technology support, have concerns about privacy and security, and sometimes technologies are just not designed for older people. The COVID-19 pandemic has emphasized the importance of online opportunities for people who are not able to go outside. To address these challenges, my research will adopt qualitative research techniques like interviews, observations, diary studies, participatory design, to explore how we can best design technologies to support older people's engagement in meaningful activities, especially for those people who are not able to attend in-person activities due to illness, mobility constraints, and geographic isolation. To make my design more effective, I'll invite people like Jack as co-designers of the digital system to ensure that older people's voices are heard in the design process. We all want our grandma, grandpa, our mom, dad, and ourselves when we grow old to be able to use fantastic technologies to attend a wide range of activities. I really hope my research can help with that, even with just a little step. Thank you very much.
All right, so thank you for your very polite applause for everybody after every single presentation. Let's do a mad, wild round of applause because they are here. And really, that's just to warm up your fingers because now you need to do some voting. So you should be able to scan this QR code. And even at home, hopefully you have access to the QR code so that you can vote about who you thought made the most wonderful presentation today. Judges, I would like to say thank you very much for your attention. It is now time for you to leave the room. I've got the names right here in front of me. Oh, I'm sorry, it's so stressful. You're all excited. I've got the names right here in front of me. It's really exciting. So first of all, congratulations to everybody. You did already win, etc., etc. You know that, but now you just want to know who actually won the competition for today. I know. Would we take some notes from the judges? Would you like to make some statements?
Are we on? Hello? Yes. Excellent. Thank you to all the presenters. We were unbelievably impressed at your presentations, and as an early career researcher myself, who's not that far from, who still remembers doing their PhD very clearly, because it was only a few years ago, I can't imagine trying to get mine down to 3 minutes. So congratulations to everyone. The standard was incredibly high. We found that sort of as we were trying to come up with the winner, it really did revolve around the smallest little things because everyone's standard was really high. One of the things I think we looked at, we're a very interdisciplinary team here, and if all of us could understand all of the words you used, you got big points. That was one of the things we looked at. So if you didn't win today, don't feel bad. It was fantastic. All of the presentations were really good, and there was certainly none that we sort of immediately was able to sort of cross off and go, well, they weren't any good. So well done, everyone, and I'll hand over to Kat to make the big announcement.
Thank you, Anthea. Okay, marvellous ones. Here we go. For everybody watching at home, this is the moment we're about to announce the People's Choice. We're going to begin with all of your choices and your choices at home, the People's Choice. Let's see if you recognise the title of the presentation first, shall we? Picture this. Insights to the future from inside the eyes, Kylie Go! Okay, goodbye. That's all right. That's okay. All right, thank you, people, for voting, and thank you for everybody at home for voting. The runner-up for the grand final of the 3-Minute Thesis 2022 is Location, Location, Location! Bridget Duckworth! Congratulations! First of all, congratulations! Well done on this beautiful presentation. Yes, that's right. I'm getting confused for a reason. I do get confused anyway. There's a lot going on. Doesn't always come out right, but I'm confused for a reason this time because the actual winner of the trophy, which I'm trying to give away, is somebody who has already received flowers because the winner is Kylie Go! There she is. Well. Kylie, no pressure, but if you did want to say something, now would be the moment to say it. Did you want to say something?
Do I? I don't know.
Just say thank you.
Just thank you. Thanks for listening to everyone. Thanks for, you guys are amazing. I was reading through the things about you guys. I was like, this is what imposter syndrome feels like. Like, you guys are amazing. I just want to hear about all your work. So, yeah, thank you.
Thank you. And you get more flowers, but that might be an overabundance to carry home. Wow. All right, so I would like to thank our wonderful organising team from the Researcher Development Unit, Alana in particular. You have done an amazing job. It's huge. It's been going on for a long time and we're really grateful for your work. Thanks to KJ for all your support and to the Researcher Development Unit where you should have all your interpersonal needs met in terms of leadership development, mentorship, as we make our way towards impact here at the University of Melbourne. For that reason, we have put on some lunch. It's really important that you spend the time to connect with one another. Be brave. Go and speak to those people who made the presentations and tell them how marvellous they are. And importantly, speak to one another. It's really so rare for us to have the opportunity in this community to hear about why we're here, what impact we all want to make in the world. We share this together. So when we sit and we break bread and we chat, this is the times that we have. So I know you're busy, and if you have to go, you have to go. But if it's possible, please talk to somebody you haven't met before and find out what kind of investigations they're involved in currently. Thank you so much for your time, everybody.